So at the start of February 2020, so just over 18 months ago, I contacted the Midlands Innovation Group to suggest that maybe it might be a good idea to develop an immunology network. This wasn't to replace the British Society for Immunology, but this was to work alongside the British Society for Immunology to try and improve the immunology research that was happening across the Midlands, and to make sure that actually people were aware that we were doing immunology research in this region. So the idea of the network is to try and bring together immunology colleagues from across the entire Midlands, not split between East and the West Midlands. It's to hopefully facilitate discussions for grant applications, and also an important hub to share technical expertise. So the network's made up of senior academic colleagues across the 8 universities that make up the Midlands Innovation. And most of those are in attendance today, and most of those will be, or several of those will be, chairing the sessions this afternoon for us. So thank you very much. So in terms of this afternoon, what's going to happen? Well, we've got 4 different sessions looking at different areas of immunology COVID-19 research. The first one is going to be antibody measurement. The second one, it will be examining cell-mediated immune responses to SARS-CoV-2 infection. The third one is long COVID. And the last one is thinking more specifically about COVID treatments. So we've given each presenter about 15 to 20 minutes for their presentation. And at the end of that, we'll have a question and answer session, depending on how much time is left in that particular session. And so please do enter any questions that you've got as the presenters are talking in the Q&A chat option at the bottom. And that enables us to hopefully share your questions with our speakers today. We've also got a quick 20-minute break partway through after the second session, so you've got a chance to go and grab another quick coffee if you need to. So I think really the last thing for me to say before we start this afternoon of exciting presentations is firstly to thank our speakers for joining us today to talk about their research. Like I say, both those that are based within the Midlands and also those that are outside our local region. And also I always at this stage have to also say a massive thank you to Alex, who basically this would not happen if we didn't have Alex supporting this network. Alex is an absolute fantastic individual that's just, you know, jumps to everything and gets everything sorted straight away. So Alex, as always, a massive thank you from me and the rest of the Midlands Innovation Immunology Network for everything that you do to make these events happen. So I'm actually going to hand over to David now, who is chairing the first session. So David Bending from the University of Birmingham. I'll hand over to you, David.
Okay, thank you, Lucy. So I'm going to be chairing, as Lucy said, the first session on antibody measurement. So I'll just give a brief background to both speakers. So first up, we've got Nina Garrett, who is R&D Director at Abingdon Health. So Nina joined Abingdon Health in 2019 and brought with her over 18 years of experience in the development and production of lateral flow rapid tests, which I think all of us are now very, very familiar with. Nina's held several senior roles, previously at British Biocell International, which is now the BBI Group, before moving to oversee Abingdon Health's assay development and technical transfer teams. So her talk will be a technical overview on the ABC-19 rapid test. And then after Nina's talk, we have Dr. Adrian Shields from University of Birmingham, who, so Adrian is a clinical lecturer at the Institute of Immunology and Immunotherapy. He's a clinical immunologist interested in understanding the mechanism that protects us from infections and how those systems fail in individuals with primary and secondary immunodeficiencies. His recent work has described risk factors for severe COVID-19 in individuals with immunodeficiencies, and also involves understanding the antibody response to SARS-CoV-2 in large cohorts of healthcare workers. And his talk will be the question, how important are antibodies in SARS-CoV-2 immunity? Insights from the Immunodeficiency Clinic. So I'd like to invite Nina Garrett to give the first talk now.
You are muted, Nina.
Just loading these slides now, apologies. There we go. Is that screen sharing okay?
Yeah, that's great.
Good stuff. And good afternoon, everybody. As I've been introduced, my name is Nina Garrett, and firstly, I'd like to say how pleased I am to be invited to talk at this event today. I'm going to be discussing in more detail a technical overview of the ABC-19 rapid test. I'll talk through the performance data we've gathered around testing patients with infection, and also review patients' response to vaccine and how we've been developing ways of monitoring antibody levels following vaccination. So my first slide is a recap of the test format. The ABC19 is a lateral flow antibody test. As mentioned, we're all very familiar with these at this point. As you may know, there are two different types of antibody tests typically available. The first uses a total IgM or A capture method, whereas the second uses an antigen test line. So the ABC19 test uses the antigen test line format and detects IgG antibodies specific to the spike protein. I think detection of IgG antibodies to the spike protein has an advantage over detection of total antibody response or antibodies to the nuclear capsid protein, as antibodies that bind to the spike interfere with the virus's ability to enter human cells. And these antibodies confirm that the patient has developed an immune response to vaccination. So testing for antibodies specific to the spike can allow far broader conclusions to be drawn regarding immunity. So just a bit more background on the test. It's currently CE marked for use by professionals. It's easy to use. You can see in the workflow that a finger stick blood sample of 5 microliter is added to the test. And then that's followed by a test solution. And the result is available after 20 minutes. Our test performance in analytical studies demonstrated a sensitivity of 98.03 and specificity of 99.56%. So looking at a little bit more detail around lateral flow and what's actually inside the plastic casing. This is a lateral flow test strip, and as I say, typically that's encased within an outer cover. So the first key area of the strip is the test line, which is coated onto the nitrocellulose membrane. And as mentioned, that's the full trimeric spike protein that we use as that reagent. The second key area is the gold conjugate, which is dried into the test strip, and that's a gold labeled signal molecule which binds IgG antibodies. The test functions by application of the sample and the test solution to the strip, after which the sample passes through a filtration pad, and that functions to remove red blood cells. The sample and matrix then flow into the conjugate pad where it reconstitutes the dried gold conjugate. The sample and conjugate then move through onto the nitrocellulose membrane, and it's here where any antibodies present to the spike protein will bind at the test line, and these bound antibodies are subsequently labelled with the gold conjugate signal molecule, resulting in a visible signal. So this is seen by the end user as a red band or test line. The sample and conjugate then continue to flow through the nitrocellulose to the control line, which will also develop as a red band and will confirm that the test has been performed correctly. The sample then finally moves into the absorbent pad noted at the end, and that assists with the flow of the test and absorbs any excess sample and conjugate that are in the system. As mentioned, the tests are really simple for the end user to interpret, as the test is positive for SARS-CoV-2 IgG antibodies if 2 lines are present, and negative if only 1 line is present. So overall, the test will confirm if a person has produced a measurable immune response to either the spike protein of the virus, and a positive result would be indicative of either previous infection with COVID or a response to vaccination. So my next slide just reviews a recent study where we've substituted different COVID variant spike proteins into our standard test line to determine where the antibodies bind to them. To evaluate this, we substituted the current test line with the full-length spike versions of the 3 main variants that we have at the minute, being Brazilian, South African, and UK. So that's alpha, beta, gamma. We've also looked briefly at Delta. However, I'm not presenting those results here at this minute. So if you're looking at these tests from left to right, number 4 is the UK, 3 is the South African, 2 is the Brazilian, and 1 is the wild type, which is a current ABC test. So the donor sample used in this example has been used in each of the tests, so it's the same sample on each. And this is from a post-AstraZeneca first dose vaccinated individual. And this is a strong responder sample. When looking at these results, each of the tests shows a signal response indicating that antibodies produced post-vaccination are binding to each of these 4 variants at the test line. We continue to review the performance of the ABC test with variants of concern, and more data will be available shortly on testing samples for infection with different variants, and so we'll be presenting that soon. So during development of the test, we observed that different test line intensities were seen for various clinical samples. And additionally, that samples with very intense lines could be diluted down to give weaker signals, which indicated that the device itself was capable of yielding a different intensity depending on the level of antibody present in that sample. To aid the recording of test line results, a scorecard was introduced to grade the test line, and this allowed comparison of a printed line of known intensity, which we will call the score, with the sample being tested. So that's represented obviously in that image on the page there. So what this does is this allows us to compare a printed line of known intensity with a known sample being tested, and this is useful for a number of reasons, as we can use it not only in the manufacture of quality control of the process at Abingdon, but it also can ensure that sort of batch to batch consistency and performance in production can be monitored and consistent. But it also means that that test line signal intensity can be assessed when evaluating samples to understand the level of test line signal response in comparison to antibody concentration. So to demonstrate this further, this slide shows an Nibsc diagnostic calibrant, which is code 20162. And this was achieved by diluting the stock, which is at 1,000 units, into negative EDTA plasma over a range of concentrations. And these were tested multiple times using 3 different lots of the ABC. And the tests were interpreted with the scorecard to yield a dose response curve. So the response for the 1,000 units per mil calibrant was at a grade 6. So I don't believe I have a pointer, but sort of midpoint of the line is that 1,000 units per mil. I hope you can see that. And we were getting a test line score of 6 on the scorecard. And when we diluted that down to 30 units per mil, it showed a really nice linear dose response relationship. And this also then allowed us to sort of extrapolate out to the highest score line of 10, which would be associated with concentrations in excess of 17,000, as outlined on the graph here. This data shows that the test does give different test line signal intensities depending on the antibody concentration. And we believe this is a useful tool for researchers investigating antibody response to SARS-CoV-2. And this scorecard is now available for research use only, and more details are available on our website if that is of interest. But yeah, we think that it's a really useful tool for being able to look at antibody response and measure it. So to follow that, we then looked at a larger panel also from Nibsc, and this is a calibrated panel which is comprised of 37 plasma samples which are known SARS-CoV-2 antibody status, and in this panel there's 23 positive and 14 negative samples. Again, the ABC19 test result confirms that an increase in test line signal intensity is seen with an increase in SARS-CoV-2 specific antibody concentration. So the next data sets that I'll show will look at how the test has been evaluated recently with vaccines and how the test is useful in detecting antibodies to vaccine. So as you know, vaccines function by stimulating the body's immune system, and the focus of the manufacturers of vaccines to date has been to produce an immune response to the spike protein of the virus. The current vaccines may use different mechanisms, but the overall generation of an immune response to the target is the overall response that's being sourced. So it's because this test detects IgG antibodies to that full spike protein, which helps identify the output of the immune system's response to vaccination or infection, that it is relevant for use. So the next slides in the presentation outline how we've monitored the response of individuals to their vaccine. A number of scenarios are described which demonstrate how an individual person's response to vaccination may depend on a number of factors. So if we look here, these are actually 2 individuals. So each graph is from a person who's had their vaccinations. So the test line signal intensities are shown following vaccination, but the blue dot on the graph represents the first vaccination time point, and the red dot on the graph represents the second vaccination. So it can be seen from this data that the test line signal response after first vaccination is low. However, it does increase after second vaccination with both patients before reducing over the following weeks. So this case study is slightly different in that the person was confirmed infected with COVID prior to vaccination. And that's obvious when considering how high the test line response is in relation to the previous slide. The highest test line signal intensity of 10 is actually seen with this patient. Interestingly, the response does not really increase following second vaccination, and is seen to decrease slightly and then level off in the following weeks. And you can see here the images to the right are the pictures of the test devices at the different time points outlined in the graph. And you can hopefully see a visible reduction from that first score, which is a 9, to the second score, which is 6 in those device pictures there. So again, this is another slide which outlines a person who was not confirmed infected with COVID. However, they did have symptoms. And as you can see from the graph, the test line signal response has increased following vaccination, again, especially when considering the results in case study 1 where there was no prior infection. Here, the signal intensity is seen to reduce slightly before showing an increase following that second vaccination. And again, the signal responses are clear in the images, that there is a definite increase from that week 1 time point over the first and second vaccination scores. So again, a clear response can be seen in that test line signal intensity. So this is the final case study, and this slide has a lot more detail to take you through. So to outline what this is showing, what is here is the results of 2 adults, KH and CH, who are living in the same household with a teenager. So each line on the graph shows the adult's test line signal response following vaccination. And following second vaccination of the 2 adults, the teenager in the household tested positive for COVID. And this is at around week 15, and that's represented by the purple arrow sort of in the middle of that graph. At this point, KH also contracted COVID, which is the blue arrow slightly to the side of the purple arrow. So as you can see, and as would be expected from the previous graphs, the red line clearly shows an increase following infection. The line of CH was not infected, and his test line and signal intensity can be seen to decrease, as seen with the previous non-infected case studies. So here, if taking case results from the previous slide in isolation, the test can clearly be seen to increase from a score of 1 at point of infection, just at that week 15, to 10 at its highest response. And interestingly, the case partner had a test line signal of 4 at the time of infection. However, he remains PCR negative. These case studies demonstrate that an individual's test line signal response can vary depending upon a number of factors, and that this is important when considering the correlation of test line signal intensity with antibody concentration. I think how antibody concentration links to immunity is an important question which also needs consideration, as this is obviously something that is being looked at as we learn more about how an individual's immunity develops following infection or vaccination, and also how it's impacted over time. So in summary, these studies demonstrate that there's an excellent case for the ABC19 test in supporting efforts to build and understand immunity. This is going to be achieved by helping to track the effectiveness of vaccination programs, identifying those who may potentially only need a single vaccine dose, tracking the development of immunity and the understanding of its spread across populations, along with identifying groups that require booster doses and those groups that have not responded to vaccination. So that completes my presentation, and thank you very much for listening.
Thank you very much, Nina, for a really interesting talk. So next up, we will have Dr. Adrian Shields, and then after that, we will take questions. But if you do have any questions, feel free to start posting them, and we'll take them after this next talk. Over to you, Adrian. I think you're still muted.
Yeah.
Yeah.
I'll just share and make this a bit bigger.
Yeah, let's share the screen.
Is that working properly?
That looks good.
Good.
Great.
So thanks, David. I'm Adrian Shields. I'm a clinical lecturer at the University of Birmingham. Specifically, I work in the clinical immunology service. That's my office on the second row down, second window from the right. We're a translational immunology unit that specialises in assay development and also runs a regional service for the diagnosis of common immunological problems and haematological malignancy across the West Midlands. I also work here at the Queen Elizabeth Hospital in Birmingham as a clinical immunologist, and my special interest is in looking after patients with immune deficiency. And obviously that population has been significantly affected by the COVID-19 pandemic. So as this session is on antibodies, the first question we should ask ourselves is why we focus on the serological response. And the answer to that is, firstly, because IgG is an end product of most immune responses. It's quite convenient and relatively easy to measure. And from the perspective of assay development, if we want to measure antibodies, the measurement of those antibodies has multiple different applications. And from my clinical perspective, antibodies are potentially replaceable in patients who lack them. So there's an immediate translational impact. In terms of what we can do with antibody assays, and the clinical immunology service has done a little bit of all of these things, we can do seroprevalence studies. We can look at correlates of protection from infection or reinfection. We can use antibodies to interrogate overall immune function, particularly in the context of natural infection and vaccination. And that's what I'm going to focus on today. And as Alex Richter has spoken here previously, you can also use these antibodies to confirm diagnoses such as PIMS-TS or long COVID. So as I said, my particular interest is in immune deficiencies, both primary immune deficiencies, in particular monogenic immune deficiencies, where single inborn errors of immunity lead to phenotypes where individuals are particularly prone to particular types of infection. But also the far more complicated, in some respects, secondary immune deficiencies, where either through someone's illness or the treatment that they receive, they suffer an immunological insult that leads them prone in the long term to further infections. And the COVID-19 pandemic has really created an extraordinary prism to look at these patients and understand a little bit more about immunity. For two reasons. Firstly, we've had an awful lot of cases in our community. And secondly, there is no pre-existing immunity to confound our assessment of what that does to a person. Just to put that in contrast, if we look at all the infections we've looked at previously, even some of our most unwell patients, those individuals who need immunoglobulin replacement, retain some humoral immunity to common bacterial and indeed viral antigens. This is some data taken from our cohort of secondary immune deficiency patients at the QE. And what we find is if you look at the blue bars, those are patients who need immunoglobulin replacement therapy. They do have protective levels of antibodies against certain pneumococcal serotypes, like common serotypes such as pneumococcal serotype 14, Haemophilus influenza B, and tetanus, despite the fact that they suffer recurrent infections with other forms of pneumococcus. This is certainly not the case with SARS-CoV-2, where there's no pre-existing immunity within our cohort. So moving on to COVID and immune deficiency. The very first question that our community needed to understand was how vulnerable patients with primary immune deficiencies, that's PID, or secondary immune deficiencies, SID, are susceptible, how vulnerable they are to COVID-19. And in order to answer that question, we have utilised the UK Primary Immune Deficiency Network, which is a network of physicians and scientists who help look after patients with immune deficiency across the UK and ask them to gather data on their particular groups of patients. And we have 326 cases of COVID-19 documented in these patients by July 2021. Now, these patients were all under the care of an immunologist. What we have found is that the case fatality rates in our patients are extraordinarily high when compared to the general population. Here we've broken down the data for common immune deficiencies, common adult immune deficiencies. CVID is common variable immune deficiency, the most common adult onset immune deficiency, primary immune deficiency, and also X-linked gamma globulinemia. And we can see case fatality rates of approaching 20% and approaching 10% in those groups of patients. In the secondary immune deficiency cohort, it's almost 30%. And when we looked at this in more detail, we found risk factors including increasing age in the primary immune deficiency patients, in keeping with the general population, but also if you are lymphopenic, before you get COVID, that is a significant risk factor for death in both primary and secondary immune deficiency patients. On average, our patients are dying 20 years younger than the general population. So this represents one of the most vulnerable cohorts in the community. The next question is, does morbidity or mortality relate to an antibody response? And this just serves as a reminder that using a sufficiently sensitive assay, over 98% of healthy individuals mount a detectable anti-spike response, anti-spike antibody response following PCR-proven natural infection. The determinants of the magnitude of that antibody response are varied and include things like age, non-white ethnicity, and increasing BMI, all of which increase the magnitude of that antibody response. This is data from healthcare workers that we've recently published, looking at what drives higher antibody responses. But when we look back at our primary and secondary immune deficiency patients, we find that only approximately 50% of them actually mount any sort of antibody response at all following natural infection. Now, this is interesting for two reasons, because some individuals get spontaneously better without any treatment at all. So we've had patients at home who've caught COVID and then they've got better and they haven't mounted an antibody response. But on the other hand, we also have patients such as this one. This was a lady who'd received a drug called rituximab, which is a CD20 depleting agent. So it ablates and induces a state of B-cell aplasia within about 3 or 4 days of administration. And she had that 34 days before contracting COVID in March 2020. What happened to this patient was that she had persistent pyrexial illness, elevated CRP, and progressive lung changes for almost 100 days throughout March, April, May, and into June of last year. This was resistant to multiple treatments. She had dexamethasone, remdesivir as an antiviral. She had recrudescence of her fever after remdesivir was stopped. The only thing that made her better was convalescent plasma. And what we did, we proved that before she got that plasma, there were no antibodies in her serum. 7 days after administration, antibodies were present. When we took those antibodies out and ran ex vivo neutralization assays, we found that they remained at a high titer effective neutralization ex vivo. Eventually, because this is passive transfusion, they do decay away. And this really tells us, and has been shown in other case reports by other groups, that some individuals will only get better and achieve viral clearance when you supplement and give them high titer neutralizing antibody products, either plasma or drugs like Regeneron. And this is also supported by national data gathered by David Lowe's group, which we've contributed to, that shows the best way to treat prolonged or relapsing remitting infection in patients with immune deficiency is with a combination of remdesivir and plasma or monoclonal antibody. So moving on to the other sort of public health strategy to prevent infection, that's vaccination. The UK has an overall strategy for looking at vaccine responses. We have studies set up in various disease cohorts, including rheumatoid arthritis, liver disease. They're all contained within the OCTIV study. Vivaldi is looking at vulnerable individuals in care homes. And I, along with Siobhan Burns and Professor Alex Richter here at the University of Birmingham, are helping run the COVID study, which is looking at antibody deficient patients. All these studies are using similar protocols and technologies to harmonize comparison between the cohorts, and we're using the pitch and siren cohort as a de facto control group. These are healthy healthcare workers. To date, we've recruited 320 patients to this study, all of whom are either on immunoglobulin replacement therapy, so they are antibody deficient, or they have a very low baseline IgG and are receiving prophylactic antibiotics because they have recurrent infections. And our outcomes here are to look at chronic viral infection and the prevalence of that, the response to vaccination, the immunological correlates of that response, and to see if we can glean any insight from patients with monogenic diseases within our cohort in terms of the immunology of immune responses. So far, we've had 6 patients develop COVID-19, either detected on study entry or have developed COVID-19 during the study. We've had 1 death in a B-cell aplasic individual who had no access to antibody-based therapeutics and had been double vaccinated. And work is really ongoing to look at viral persistence and evolution in this cohort. In terms of the serological response to vaccination, we know that healthy individuals from the pitch cohort, all of them mount a serological response to 2 doses of vaccine. In our COVID cohort, that's around 55%. And you can see a group of patients right down at the bottom here in the serum. These are patients with either XLA or induced B-cell aplasia who mount absolutely no response to vaccination at all. We've had a few patients who we've managed to sample before and then after their second dose of vaccine. Numbers are a little bit small. We do have more, we just haven't analysed all the data yet. But the take-home message here is that the second dose does boost overall seropositivity, and we achieved the 52% seropositivity after that second dose. And this is important because booster immunisations may be helpful to some of these patients. The real critical question is when to time those boosters. And it's particularly important in patients who've had B-cell depleting agents. And to try and answer the question of when to immunise a patient who's had rituximab or another B-cell depleting agent before, we've recruited 118 patients across the West Midlands who've had previous exposure to anti-CD20 B-cell depletion for any disease. We've got 36 rheumatology patients and 80 hemato-oncology patients, and tested their response to vaccination after their second dose. We find that vaccination post-vaccine seropositivity is almost 70% in rheumatology patients and 60% in hemato-oncology patients. We also found there was no difference between the magnitude of antibody responses or overall seropositivity between the 2 vaccines which have formed the bulk of the UK vaccination effort, AstraZeneca and Pfizer. But interestingly, we did find that shorter intervals between the 2 doses significantly improved vaccine responsiveness in hematology patients, both in terms of seropositivity and the magnitude of that response, with a similar trend which was non-significant in the rheumatology patients. The major determinant in terms of cellular populations of the size in terms of seropositivity post-vaccine response was whether or not you had B cells. That's entirely predictable for a set of patients who've all had B-cell depletion previously. But what's important from a public health perspective is that the rates of seropositivity following vaccination is pretty poor in the first 6 months, and then increases significantly in the 7 to 12 month window or 12 to 18 month window following last B-cell depletion. And this is an important message for people receiving that. And we can look at that in more detail in terms of B cell reconstitution following B cell depletion. We see that there is very few B cells in the circulating blood up to 6 months, very low levels of vaccine responsiveness, which increase rapidly in the 7 to 12 month window. But what's also interesting is that we find a cohort of patients, particularly hematology patients, who, despite having distant B cell depletion, on vaccine non-responsive in the late phase. We don't know what the mechanism of that is, but it does illustrate that probably secondary immune deficiency is dependent on what initial disease you had, and also that there may be different mechanisms depending on time from previous B cell depletion. So in conclusion, patients with primary and secondary immune deficiency are extremely clinically vulnerable to COVID-19. What differentiates those individuals who can control viral infection in the absence of humoral immunity from those who cannot is not clear, and is something we're investigating with great interest at the moment. The response to vaccination in antibody deficiency overall is about 55%, and boosters may be helpful to these individuals. And following B cell depletion, vaccine responsiveness returns after 6 months in most, but not all patients. Why is all this important? Well, the reason why it's important is because individuals, particularly those with blood cancers, but also those being treated for other inflammatory and autoimmune diseases, are living with the side effects of novel treatments that have immunological effects for longer. We're using all sorts of different strategies to treat these diseases, either in combination or in isolation or sequentially, and they do have cumulative immunological toxicity. We can't yet regrow an immune system, and unfortunately, there have been no new treatments for primary or secondary immune deficiency in 70 years. We are still using immunoglobulin, which was developed back in the 1950s. And unfortunately, we've all but run out of immunoglobulin globally because of a lack of plasma donation during the COVID-19 pandemic. So we urgently need to think about how to manage these patients and manage the potential long-term toxicities of these treatments. This is a huge collaborative effort nationally through all sorts of different groups of individuals, all of our colleagues in clinical immunology across the UK. The programme of research has been headed up by Professor Alex Richter and Professor Siobhan Burns, and we had funding from UKRI and the Association of Clinical Biochemistry and Laboratory Medicine.
Great, thank you. Thank you, Adrian, for a great talk. So looks like we have plenty of time for questions in this Q&A. I guess while people are thinking of them, I've got at least one question for each of you. Maybe as Adrian, you've just finished, I'll ask you first. So you mentioned that some patients have spontaneous viral clearance. I don't think you know entirely why, but I wonder if you have some ideas. Is it related to sort of potential T cell or NK cell immunity, or is it related to maybe the initial viral loads? Like, have you been able to look at like PCR threshold, CT values of people that spontaneously clear versus those that haven't?
Yeah, so we have looked at viral loads in the small number of patients who've been recruited to COVID. We don't have very many because we really don't want our patients to get it, to be perfectly honest. No. They appear to be, the CT values appear to be pretty low actually, and it doesn't seem to have any influence on the sort of wellness of the patient and sort of the symptoms that they experience. We've had patients who've remained positive for our longest running patient in COVID at least is 7 months of positivity. And they've had viral loads, CT values that run at about 25, which is, you'd expect that only to be sort of, if you're coming into a hospital, most of them run about 20. So it's not that much different, really. In terms of what controls those individuals or what differentiates those individuals, there was some work done by a group of individuals over in the States in hemato-oncology patients, and they attributed it mainly to, or they had patients who control the virus in the absence of humoral immunity, and it was the quality of the CD8 response initially. Okay. But we've not yet looked at that in further detail.
Great. Okay. And I guess bring Nina into the question. I think I probably Adrian's done that too. A lot of your work's done based on blood-based, isn't it, and antibody responses. But I wonder if there's been much that's been looked at in terms of antibodies in saliva and the potential importance of antibodies in sort of coming off the mucosal tissues in a way. I don't know, Nina, if you've tested saliva in your systems.
Yeah, so our system has been designed specifically to work with whole blood or plasma serum samples. Again, we very much designed this as a use case for that sample matrix. I think there are tests out there that do test in the saliva format and the saliva matrix. And again, they're going to be having a slightly different use case for the end user. So as a investigational piece, we haven't actually looked at saliva, but of course that's possible in the lateral flow format.
Yeah, okay.
We've looked at saliva. So as part of one of the research projects we set up was to basically try and understand the occupational risk in dentists. And of course, dentists are particularly interested in saliva. So they volunteered salivary samples. We collected about 3,000 salivary samples after natural infection and then after the first dose of Pfizer, which they received in January of this year. What we found was that it's relatively straightforward to measure IgG, and it's relatively straightforward to measure IgM. But when we started trying to measure IgA, which is possibly of greater interest in terms of mucosal immunity, it was very, very difficult to actually set a sensible threshold on our assays and achieve any sensible specificity of the assay. And we're still working on it. We've done amplification steps. We've tried to absorb out sort of other factors that may be interfering with the assay, but it's been difficult. And when we're measuring the IgG, we think that at least some of it is just translocation from the blood into gingival clavicular fluid as well. So it's not as straightforward as it seems.
Sure. We've got a question in the chat. So Carl Edwards asked, have you tried mass spec for detection of viral loads in plasma? So I think that's
no.
No. Okay.
No.
Great. Okay. Someone got Lucy. You've got your hand up.
Are you okay if I ask a question?
Yeah.
Okay, so I was just going to ask Nina, and I don't know if I saw the graph correctly or not, but it looked like the antibody response reduced quite dramatically after the second vaccination, following the 8 weeks post-second vaccination. I was just surprised to see that drop off quite so quickly. Was that what you were expecting, and does that seem to match with other literature?
I think obviously we're in a situation now where boosters are being talked about and antibody waning over time is being looked at. I think from the information that we have, an individual's response is obviously based on a number of factors, as we showed, pre-COVID infection or COVID infection between the two vaccines will give you a very different shape of that antibody response and then drop off over time. So I think that's what we're still finding out quite a lot about. But yeah, I think, seeing those two graphs, they obviously did decline over time. Post-second dose. Yeah.
Is that okay? And Ella, you've got a question as well, hand up.
I think it might be a for Adrian, but if Nina has something to add, that would be appreciated. So basically, the antibody response was more or less similar after vaccination to those who actually contracted COVID among the immune deficient patients. Around 50% responded. Do you have any insight on the level of cytokines such as IL-6 or IL-10 in these individuals? Do we see different patterns in their plasma level? I appreciate any comments. That would be nice.
Yeah. So the patients who did respond, I mean, we've got survivor bias here. So we're looking at a cohort of patients. So we've only measured the antibodies following natural infection in a small number of individuals so far, and those are the individuals who've survived. In common variable immune deficiency, almost all of them have mounted an antibody response. What's become apparent is that it doesn't last very long, so it decays away. And when we look at the course of the natural history of their disease, again, small numbers, but it seems like, to give you an example, we had a patient who was pyrexial for about 3.5 weeks, and then suddenly it all stopped. He just turned off. He didn't have any additional treatment, but what we think has happened there is that he probably had quite a low frequency of antigen-specific sort of B cells and T cells, eventually found the right one, it all clicked together, and then made some antibody, cleared the virus, but they're just not long-lived because of the nature of the disease, the underlying disease. In relation to your first question, we haven't looked at cytokines or IL-6 or IL-10 in any of those patients yet. It is quite a heterogeneous group of individuals who we're looking at in terms of primary immune deficiency. We've got the samples. We might look at it in time.
Great.
Thank you. Fascinating.
So I
Nina,
guess,
do you want to comment or no?
Nina, did you have anything that you wanted to add to that?
No, I think that Adrian covered that side of things. I think that, yeah, from the lateral flow, we wouldn't be able to have that.
Great. Okay.
Cheers.
So I've got another question that I'd written down. I think sort of related to what Adrian was talking about. So am I right now that it's antibody cocktails are now approved for use in some of these patients in the UK? Is that correct? And have you started doing that? Because I guess the concern here is with people with such long viral persistence, and you're looking at the evolution, is the development of variants, particularly in response maybe to convalescent plasma, where you may be driving viral escape away from that. And so what's the concerns around that, and what treatment strategies? Because I think AstraZeneca has a double monoclonal antibody, and there's another one that was recently approved that seems to have reasonable efficacy. So are those now being allowed to be used on the NHS, and is that sort of your frontline treatment that you're hoping for with these patients?
Yeah. So in terms of when the first antibody-based therapeutic that was thought about was using convalescent plasma. That was used in the recovery trial. It was shown not to have any great benefit in the general population, and all of that plasma stock has now been parked to one side and no one else is using that. The other antibody-based therapeutics that have been tried and shown not to work in the general population are hyperimmune serum. So we sort of concentrated down high titer donors. That doesn't work in the general population either, and that's been completely parked as well. So we're left with just the monoclonal antibodies, where the pharmacokinetics and the pharmacodynamics of the product can be thought of in a little bit more of a standardized way. Regeneron is the one that has been recently approved by the MRHA. Quite how it's going to be used within NHSE, the NHS more widely, is still under debate. It looks like at this stage, in order to access it, you will have to be in hospital on oxygen. Now, obviously, there are lots of different ways you can use monoclonals. You can use them as sort of post-exposure prophylaxis, outright prophylaxis, or as a treatment. And it looks like the way things are going, they might just be used as a treatment at this stage. But we're hoping, and we've actually got some money to run a trial of the GSK monoclonal as an outright prophylactic agent, which I think is probably the way forward. It's certainly what the guys in the States are doing to try and protect their very, very vulnerable individuals.
So are there I'm aware of the trials underway to try and increase the longevity of the IgG in the plasma, so you could give it almost, instead of having a vaccine, having a shot of it each year of a cocktail.
Yeah.
But I guess how we control HIV with 3 different inhibitors so that we don't generate variants that can get around.
So there are differences in the half-lives of the monoclonal antibodies, and the one that will be going into trial for the vulnerable patients has a slightly longer half-life, which is, I think, a good thing. With respect to inducing sort of escape mutants in patients who are immune deficient, I don't think we know quite whether that will or will not be a significant problem. The only way we will find out is to sequence the virus in patients who've, I mean, we've got patients who've had it for a very long time, so it will be optimally, we think, optimally adapted to them, and we'll see whether it has significantly changed. And we don't have that data yet. A lot of the hypotheses that it's going to be a problem have come from either very immune deficient patients, and in very small numbers as well, sort of N=1 case studies. It's difficult to infer much from that, I think.
Yeah, okay. So I think Ella's put her hand down. We got someone. Ella, did you want to ask a question? Or your hand popped back up?
Yeah, about the stratification, these patients with immune deficiency, are they diverse from, you know, ethnicity point of view, age? Particularly, do you have like immune deficient children that might respond differently? How was your experience to share? Thank you.
So kids with immune deficiency, obviously there's a spectrum, and immune deficiency sort of all gets, in terms of primary immune deficiencies, there are about 450 monogenic disorders plus some polygenic stuff which we don't completely understand. So there's an awful lot of different things that can go wrong with the immune system. What is pretty clear from the data so far is that regardless of whatever you've got, the children seem to be less affected and have less morbidity and mortality associated with SARS-CoV-2 infection, even to the point where, you know, some kids who've had severe combined immune deficiency and have caught the virus shortly after transplant, they've been okay, which is very reassuring from the pediatric point of view. We've also had kids who've got XLA, X-linked gamma globulinemia, so BTK deficiency, complete absence of B cells, who've been fine. They've had the virus. They seem to have cleared it on the basis of non-humoral immunity. As you look at the cohort overall, and as they get older, we find that there is a cutoff at about the age of 40, where suddenly the infection fatality and case fatality rates significantly increase and sort of completely depart from that of the general population. So if you think of the general population, sort of you're getting significant increases in fatality rates above the age of 65. It's happening 20 to 25 years younger in this cohort of immune deficiency patients. Now, what's driving that isn't entirely clear. There are lots of hypotheses. Some people have attributed it purely to immunodeficiency associated comorbidities like lung disease, and certainly that is probably a contributory factor, but equally the presence of lymphopenia suggests that there are also some patients who are have some sort of intrinsic immune system differences that may also be contributing to that failure to mount a successful immune response early to the virus.
Thank you.
Great. I think if there are no more questions, when we're kind of on time, I'll pass on to Lucy to share the next session.